there is a very rare kind of cancer called malignant peripheral nerve sheath tumor that happens in peripheral nerves. You guys know where it is? Peripheral nerves? What are peripheral nerves? Physiology. Learn about yourself tomorrow. Okay? You guys like this funny way I speak, right? Sometimes. Okay. My kids, when I start saying this, my kids say, Papa, shut up. They go away. Okay? Anyways, that I'm saying this so that you can remember things. All right? I can speak very normally too, but that will be very boring. Wouldn't it be? Right. Okay. This particular cancer called malignant peripheral nerve sheath tumor happens in those. Remember a picture of a British actor I showed you guys on the first day? Those people who have NF1 mutation get malignant peripheral nerve sheath tumor. Okay? And I actually work on those. We published a lot of papers on these. Not a lot of papers, some papers. But NF1 mutation, NF1 gene deletion happens in some of those patients. Okay? Right next door to NF1 gene is a gene called SUSY12. What is SUSY12? Susie, not Miss Susie, SUSY12, SUZ12. This gene right here. When NF1 is deleted, co-deleted with it is SUSY12 gene a lot of times. 70% of such cancers have SUSY12 deletion or EEDG deletion. That's right next door. Okay? So what happens in those patients? Those patients do not have the major repressor complex. This complex represses many, many, many hundreds and thousands of genes in your body. How cool is that? Is that cool? So major repressor complex in your body is gone in those cancers. But those people make a lot of very aggressive cancers. There are 1,500 people who get those cancers only in United States. Probably across the world, maybe a couple of thousand people. But it's so cool. I found it so cool. I didn't know about this till I started my lab, and a next door neighbor professor was working on it. I was like, "Woo, I love this shit," because SUSY12, I could not comprehend how PRC2 complex deletion will make a cell survive. EZH2 deleted mice do not survive, but EZH2 polycomb deleted cells go on to excessively proliferate and make cancer. What is cancer? Big tissue formed in places where it should not be formed, right? So I'm just telling you the significance of these things again. I just want to keep you interested rather than saying SUSY12 PRC2 does H3K27 trimethylation. That's a major repressor complex in the body, but that does not have any meaning. That does have a lot of meaning for those 1,500 patients who get this every year. Okay? All right. Ring 1B is the other catalytic unit of the complex called PRC1. PRC1 and PRC2 work together to silence genes. Okay? And I'll show you why in a second. Ring 1B is a histone ubiquitinase. It's a histone H3 ubiquitin ligase. It does histone H2A lysine 119 ubiquitination is done by ring 1B. Okay? Cool? So there are two marks that are made by two complexes that are both called polycomb. One does ubiquitination, one does H3K27 trimethylation. They act together to silence things. They act together. Histone code, remember? Okay? All right? So those is polycomb complexes. So no wonder people found polycomb repressor complex 1 and polycomb repressor complex 2 in the screens they did for suppressing the same phenotype. Of course. They're in the same pathway. They do the same thing. They exhibit the same phenotype. All right? Are we cool with that? Did you get it? Okay. And then there are other groups, H3K4 methyltransferases, that are called trithorax group of proteins. What do? They do the opposite function. I told you yesterday, H3K4 methylation, what does it do? Activates genes, right? H3K4 monomethylation does what? It marks enhancers, I told you guys yesterday. Do you remember? H3K4 monomethylation only, and dimethylation, mono and di. Does enhancer marking. H3K4 trimethylation, on the other hand, is always there on the transcription start site of genes that are to be activated. I told you this yesterday as well. Because H3K4 trimethyl does what? Yeah, it recruits a PhD. PhD recruits that. PhD helps you recruit Tata binding proteins and the QL, right? So they do opposite functions. K27 trimethyl is used to suppress the gene. K4 trimethylation is used to activate the gene. Some genes in our body, actually our cells, have both of them together. An activation mark and a repression mark all together. That's confusing, right? They can be there together, K4, K27, they're not on the same residue. There are two together and one nucleosome. Some nucleosomes contain both marks together. Funnily enough, repressive activation. It's called bivalent promoters, bivalent genes. Okay? Why would somebody want to do that? Can you guess? Let's be a detective for a minute. Why would the cell want to mark certain genes with bivalency, where one is repressive, one is activating? Why would cell want to confuse itself? Guess, any guesses? My intelligent folks.
Cell want either to express or not to express that gene, so it make both the marks on the gene. In either condition, it will express or not express.
Okay, somewhat of a good answer. What else? Where would you want to mark it? Which genes would you want to mark with bivalent marks? Genes where?
For hormones.
For hormones.
If there is an overexpression of the we just need to have a negative feedback loop.
Okay. All right. Bivalency is usually not found in hormones. Okay. Bivalency is found on genes in stem cells, on genes that where you can have either function in your daughter cell. I'm a stem cell and I want to make a cell type A and cell type B from both pluripotent cell. Okay. I don't know which one. So I can have certain genes marked with this bivalency. So when I go to the next one, like A, I'm just going to remove my repressive mark for that to be activated. On the other gene, I will just remove my activating mark for it to be repressed. So a lot of genes, bivalency genes formed on genes which are pluripotent in pluripotent cells, meant for developmental specificity eventually. Okay. All right. Does that make sense to you? Let's just make it easy. Cells like, boom, okay, I'm going to have both. I'm just going to remove one here. I'm going to remove other one here. That's all. So it's really, really very important genes that are developmentally important to specify the phenotype actually are marked by bivalency. There was a paper by a guy called Brad Bunstein in 2004 or 8, something like that. There's a Cell paper, and he's known for this. He's become the department head for Dana-Farber Cancer Institute, the prime cancer institute in Harvard University. You do great discoveries, you do great things, you get money and fame. Okay? All right? All right, on the bottom are MLL and SET protein. SET protein is a key enzymatic domain that will mark H3K4 methylation. Bottom line. So any enzyme that has K-SET domain actually are usually marking methylations. Okay? Cool? SET domain is a domain. SET domain. S-E-T, SET domain. Okay? So MLL34, MLL12, MLL5, these are all enzymes that do H3K4 function, and they are actually the primary things that mark gene for activation. So very, 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 very important enzymes, of course, right? MLL3 and MLL4 are found to be mutated in about 20+% of cancers, 10 to 15% of cancers, about that, 15 or so, I think. My lab studies MLL3, MLL4, we do make a lot of mouse models of this, and we've shown a lot of important things. So there are patients who come to clinic with 15 to 20% of patients with MLL3 or MLL4 mutations. So very important in cancer. There's a syndrome called Kabuki syndrome where you have developmental disorders where MLL4 gene is mutated. That's solely defined by that. In fact, many neurodevelopmental disorders or very developmental disorders are totally caused by epigenetic genes. Just one mutation in one gene. That's it. They are monogenic disorders. Okay, so they're obviously not lethal. They're not, patients are born with it. The kids go on to live a decent life, but they have abnormalities in their development. They have growth delays, they have mental retardation, and a lot of those. So there are lots of syndromes like this. You guys hear about syndromes, right? Genetics, no genetics yet. Syndromes are amazing. You can cure them very easily because one gene problem. Cancer is a multi-gene, multivariant problem, so very difficult to cure. But these neurodevelopmental or developmental disorders actually typically have one gene problems. Very easy. Look for, not very easy, but of course much easier. None of this is easy. But there are patients who are born with this. There is a good market. People have not tapped that market yet. Okay. I myself have 2 companies, by the way. So therefore I'm actually a little bit entrepreneurial in my mind. So I will keep on telling you market and where you can invest. One of my companies is in Bangalore. I told you, right? I have a microbiome company. And I have nothing to do with microbiome. I'm not a born microbiologist. I was trained epigeneticist. That should tell you can do anything you want. My other company is on immunology. I was not trained as an immunologist in my PhD or anywhere. In 2019, I thought immunology is great. I want to learn and do it. So I said, okay, I can do it. I wasn't born, right, with that saying, oh, immunologist. You're not born to be a plant biologist if you do plant now, right? You could tomorrow be a cancer biologist for all you know. You don't need to be instructional biologist forever. It's a good thing to be, but you don't have to be. You have to trust yourself and say, okay, my PhD, my MSc is all about me learning how to learn something new, how to think about a problem, how to solve a problem. That's all PhD teaches you. PhD only teaches you how to solve a problem, a biological problem. That's it. Sudoku, it's playing Sudoku, right? Tomorrow I could play another one. As long as I learn how to solve a problem, I can solve any problem. So that's what I believe in. That's what most scientists believe in. That's what you should believe in yourself. So I said, okay, I'm trained in epigenetics for 20 years. 20 plus years, but I can do immunology. It's actually fun to learn something new, right? So I collaborated with an immunologist and so I said, teach me immunology. So I collaborate with him. We work together. We filed our patents. We made our company in 6 years' time. We are first in class clinical trial for something. So we're trying to find. My company has no money right now. We're trying to raise money, but nonetheless, you could do this, right? You get that? You can do anything you want. And I'm sure you have lots of great examples of lots of great people who've done anything in their one. By the way, on that lesson, I should give this lesson to you guys. I would have given all my learnings in last 45 years of my life to you guys by the time I'm done with this course. I read somewhere, and it was a very interesting perspective. You guys are likely going to be living much farther than 80 or 85, right? You believe that that's going to happen? If you believe in biomedical science growth, and if you look at the science growth in last 10 years, okay, let's take a step back. Let's think about IT. When were the computers there? When did you guys get hold of computers? Did you ask your parents when did they see their first computer? I got my first computer when I was in IIT in 3rd year, right? When did most people start to get computers? Like the most computer science actually growth has happened in last 2 to 3 decades, 2 decades, 25 years or so, right? Remember the Y2K problem? You're not born then. What am I talking about? But anyways, it's all then, right? I had my first cell phone in 2008, right? You guys are lucky, you have everything. But nonetheless, what I'm trying to tell you is that most of the growth has exponentially happened in last 20 years. And remember, winter's coming, right? AI is here. AGI is supposed to come here next year or two or in 5 years, whoever you believe in. What is AGI, by the way? Do you guys know? You guys don't know what is AGI? AGI is artificial general intelligence when it start to beat humans. Right? You know this, right? So Elon Musk predicts AGI is 2 years away or less than 2 years away. That's why I say winter's coming, guys. Okay? Do you guys, have you seen Game of the Thrones? You guys have not seen Game of the Thrones? Oh man, you're all adults. You should go see Game of the Thrones. Amazing series. What an amazing series. That's one of some few things. I don't watch a lot of TV or thing, but this is something I watched during COVID, and I was blown away by the creativity of the creators. It was so brilliant. Brilliant creators. Look at Game of the Thrones. Winter's coming, that's why I say. AI is coming, guys. AI is here. Next 5 years, your life's going to be changing. At least predicted to. It may be all hoax, but that's what people believe in. That's why world is investing. All companies that are getting money now are AI. All investors are investing in AI. The whole world is talking about AI, isn't it? You got to get out of Bilaspur and think of something else. Think of what's the world's going to change in a way that you will not be able to believe. That's at least predicted. Okay. So what I'm trying to tell you is the law, and this can happen, right? Well, as growth has happened in the last 10 and 20 years. AI is going to come if it is all true that people suggest. Let's hope it is. It might take 10 rather than 5 years, but it's all going to happen. AGI will be here. Robots will take over a lot of jobs that humans have, but you will still be humans. You have to believe in your neural network here, right? Believe in it and do things, new things. Think about it and do new things, right? What did I get into AI, by the way? Because I was talking about medical science, the growth in medical science, about your lifespan. Yes. So the whole growth has happened. All of that I'm teaching you is in last 20 years. When I started being a PhD student in 2002, we only knew histone acetylation. We were just beginning to learn about histone methylation. All this histone demethylases I showed you happened in front of me. I discovered a DNA demethylase. I'll tell you in a second, after lunch. It just happened 10, 15 years ago. Only 10, 15 years ago. Only 10, 15 years ago. We're making inhibitors now. There is AlphaFold, who got a Nobel Prize last year, isn't it? AI is changing the way we design drugs. Okay? You will be living more than 85 years, most probably. Because in next 5 to 10 or 15 to 20 years, there'll be drugs coming through. Where do you think Elon Musk and what is the Amazon's founder, Jeff Bezos, invest? Where do they invest their money? They have billions of dollars. They don't eat that much, right? They actually eat less than, like, they eat all sabzi and sabzi and sabzi and sabzi, no roti and no rice, right? That's what they do. But where do they invest their money? Huh?
In the Bitcoin share.
They invest. What do they not have? Think about it. What do these guys not have? All the billionaires of the world, what do they not have? I told you there's only one thing that is common between you and Elon Musk. Time, 24 hours. Where do they invest their investments? A majority of their investments are in AI, of course, but a lot of their investments are in improving the lifespan, because they all want to live longer, because they want to make more money. Man is greedy. No, that's not why. But everybody wants to live longer. Everybody's got only 24 hours in a day, right? So you are going to live longer because the billionaire is investing in the drugs to be made for living longer. That will be available to you. I'll be dead, but you will not be, right? So why am I saying any of this, by the way? I'm saying I should be teaching you epigenetics. I'm teaching you something else. Why it is important is because when you think of your careers, plan for 100 years, not 70. That's why it's important. So I read an article and it blew my mind. I was like, oh yeah, he's right. So most people like your age, and India has the most young people, so young people will come from India and Asia. This is where the birth rate is highest. This is where people will come from. This is you guys, your kids. Think 15 years, 20 years, 50 years down the line. Those kids, your kids, will likely live for 100 plus years. A lot of them, not all of them. Okay? So what should you do? Don't think of yourself as a one-trick pony. Don't think of yourself as somebody who's only doing biotech. You might as well be doing AI. You might as well be doing Bitcoins. You might be doing whatever else. Have more hobbies. Do have more profession. So the article that I was reading was all about you should always plan for second profession or third profession, because you will obviously not want to do the same thing for 100 years. So post 70, if you live 30 more years, you would just not be wanting to be roaming around the world, right? Some of you want to roam around the world. That happens in 2 months. After 3 months, all mountains are mountains, all seas are seas. Doesn't change. The food is the same, right? I've seen decent amount of world. I haven't seen all of it, but after 10 days, it feels like same mountain, same ocean. What triggers your imagination is your brain. Stimulation is your brain. Okay, that's the humans we are. Our neural network here is what will keep going. Therefore, think of other things. Think of things that we will be able to do. So invest in your hobbies. Invest in something else. Right? If you make $1 trillion, my friend, there's nothing much you're going to be doing. There's more to it. There's more to making money. There's more to the profession. Likely, I mean, if you're all predicting to be right, then I want you to think that you're going to have 100 years of life and plan for it. And your kids may have 120. Okay? Anyways, epigenetics, by the way, has a huge component in aging. You know that, right? Aging is dictated by epigenetics. What is not dictated by epigenetics? That's why this course is so important. That's why you should pay attention and not sleep, guys. Some of you are sleeping and looking at your YouTube phones. Okay, anyways. So, okay, let's go back. Let's go back to the real world. That was imagination. But I like it. It's fun, right? Has anybody made you think differently like that? Has anybody made you think that you can have 2 jobs in your life? 2 good jobs for many, many, many years? You should think that way, actually. It'll be fun. Okay? Anyways, so polycomb and trithorax group of proteins have 2 opposing activities. We talked about this. One has K4 methylation, one has K27 methylation, right? It's opposite to each other. Some things have bivalency, they go on. So this is just a summary table I put up from there. PRC1 has multiple subunits, PRC2 does too, and then trithorax is different. Set is always a fight, or it's always a competition between polycomb and trithorax. And whoever wins makes the transcription happen in their way, and the genes get expressed that way. It's very systematic in your body that makes you who you are, makes you a normal human being. If this doesn't go right, you become an abnormal human being. Okay? All right? Good. How does PRC2 and 1 work together? Look at this. PRC2 binds to this K27 trimethylates. It stimulates this enzyme. PRC1 comes, binds to H2A K118. It silences by opposing the K4 methylation. It silences by compacting chromatin. So it puts the compactor with it. PRC1 has that activity. And this H2A cubanation prevents elongation by, again, suppressing the other mark, like H2A K119 suppresses K36 or K79 methylation. K79 methylation doesn't happen, so the RNA polymerase cannot go through. I didn't teach you elongation and histone modifications, I know, but I'm just telling you, you can read about it, okay? I mean, I just hate to tell you every single detail because it's not useful to you anyway. You're not gonna apply that tomorrow. My job is to teach you principles of, fight, principles of competition, principles of how things work. I don't need to teach you the details of it. AI will tell you that. If you just Google it, you'll find it, okay? The point is there is competition, and the PRC H3K27 trimethyl happens. K4 will not happen, and this will not work. When this works, then this will not work, and therefore there's a competition between polychrome and trithorax. So whoever wins will do what their job is. ASH1 again is a set domain protein, will mark the methylation, so that's an activating protein that will recruit CBP-P300. CBP does histone acetylation, so that will make the code that we talked about earlier. Acetylation and methylation together will activate the gene transcription that will again inhibit the PRC activity and will suppress. Okay, so activation will suppress that, and the other one will suppress this. Okay, just molecular interactions between the two things. All right, are we good? They will suppress elongation, they'll suppress initiation, so there'll be no more transcription, no more polycomb going through. You can read this slide carefully when you want. Okay. In America, they teach you not to put a lot of stuff on the slides. In India, they put a lot of stuff on the slides. That's a very different way of teaching. And when I have lots of stuff on my slide, I don't teach well. But I tell you that there's information you can go read. You're all educated people, your PhDs and MSCs, you can go read and figure it out yourself. Okay. All right. Polycomb-mediated gene suppression happens by holding poised RNA polymerase. I told you poising happens before elongation goes, right? It poises that by by just putting K27 trimethyl. It doesn't allow the RNA polymerase to get phosphorylated, so RNA polymerase is poised. That's an abortive transcription. It does not go along, and the RNA is not made. Okay, that's how K27 trimethylation works. I told you chromatin compaction happens. PRC1 goes on, compacts the chromatin right around the transcription start site or in the gene body, and the RNA polymerase cannot go anymore. That's another mechanism how it works, and that's just that. That's how they work. This is important. So how does the heritability happen? Remember the first day I said epigenetics, genetics, heritable. Cool. Heritability is the concept. Heritability has been bothering scientists for many years. How do histone modifications become heritable? We'll talk about DNA methylation in a bit, like after the lunch, but DNA methylation is easy to be heritable. I'll show you that in a second. But basically what happens is when the replication happens, you guys know replication forks? Replication fork has PCNA. Have you heard of PCNA? A protein, a circular solenoid protein that goes through, for which this replication fork goes through. That actually is at the forefront of replication fork. Do you guys know this or no? How many of you know this? If you don't know that, raise your hand. Good. Replication processes are important for study. I'm not going to teach you replication again. You can go read about it. But in replication fork, PCN is a protein that leads the replication fork. And I'm not an expert in it, so I don't remember. I probably studied it when you guys studied it. But PCNA recruits an enzyme called DNA methyltransferase, DNMT1. DNMT1 will go and methylate the DNA along with the replication fork. So it will replicate the methylation pattern. That's a very simple mechanism of how DNA methylation gets inherited. Okay? We'll talk about it again after lunch. But histone modifications are much more difficult to replicate, right? Because a protein, what gets through from one cell to another cell? Chromosomes. How do histone gets modified, gets repeated in the next strand that is newly synthesized? When you're synthesizing the strand, putting a methylation on a piece of cytosine is much more easier than it is putting in a protein, because there's no protein, right? Or there is protein. So heritability is a big problem in histone modifications, because they're not, when the replication fork is going through, it's not like they are all histones, same things around, right? Because new strand does not have histones. New strand needs to get new histones on it, right? Yes or no? You get it, right? I replicate 1 into 2, I need to have new histones around me, right? But if I have DNA and DNA was methylated as I was making 2, then it's easy. That can be done as long as my enzyme is tracking with my replication fork. Simple covalent modification. Is that clear? Can you guys imagine this in your head or no? Can you? Yes. I mean, I can imagine, but I don't know if you can. Okay. So heritability of histone modification patterns is a big, big challenge. Okay. So what people have now figured out is that the parental chromatin has this increased PRC1 activity. PRC1 actually bridges to the new chromatin. Okay, you see the bridging here happen in the new. So PRC1 is the first one that goes through. It marks the ubiquitination mark. Then it puts on the K20. It recruits PRC2 because PRC2 can bind to the PRC1 elements and go on and make the K27 trimethyl. That's the first layer of heritability that goes on. That happens on the repressive marks. In the active things, this is, I think, not very well worked out, and therefore you see a lot of question mark here. You see this question mark, question mark. So in the repressive strand, we know how this works. There are also polycomb seeding elements where PRC2 and PRC1 bind. So there's multiple mechanisms of this. Again, this is not completely sorted out yet. Scientists are still debating on this as to how this works. But there's what people have figured out thus far, and in the most latest research, is PRC1 is the major thing that actually gets recruited first as part of the replication machinery, and then goes on and recruits K27 trimethyl. Okay. So we'll leave it that. I'll come back to that later on. Actually, I want to come back to this at some point. But I was telling you, histone modification heritability is a big challenge in the field. Still, people don't completely understand that. Okay, all right. We talked about histone modifications in multiple ways. Multiple histone modifications. I showed you this slide yesterday. You guys get this? One nucleosome, one small baby nucleosome contains many modifications on it, right? Does not mean all of these modifications are on the same nucleosome, right? This is just a depiction. But a subset of this are together. And it becomes complex because there is two copies of the same thing. So one, you become H3K4 and something else. In other, you don't. Same way. You can have different patterns on the same nucleosome, right? In two different, and that will confuse the audience. Who's our audience? The readers. The readers wouldn't know which one, where, what, right? So there are a lot of things built into this. Now, as scientists, again, we still don't understand how these two tails, if they're differentially modified, how does that get interpreted? But that layer of complexity still needs to be figured out. Okay. Therefore, epigenetics question for people to figure out as we go along. But more importantly, these patterns that I'm showing you have very unique binding patterns. And why am I showing you any of this is because now we're going to go into the methods of mapping these chromatin marks on the cells. And then I'm going to go into teaching you some bioinformatics. Do you guys want to learn bioinformatics? Not really. Okay. I'm not a bioinformaticist. But we'll teach you something, okay? Something that could be interesting.
At the end of the class.
Yes, you did. No, it's not the same thing. I put this in the morning. I thought this will be easier for you to read than that one. But I'll come to that diagram also later. I'm just trying to tell you the patterns. I'll come to patterns. This is next. We have 1 hour until 1:15. So I have to finish at 1:15 right now. So I want to go through a few things. You'll see. But I'm going to try to get you to think about how to map these histone modifications on a genome to have the meaning of this class, epigenomes, right? We need to understand epigenome. This is where epigenome is. So this is the crux of it. That's why I'm taking 2 days rather than 1 to teach you this, okay? And we'll come to DNA methylation, then we'll go to the other stuff and other stuff and other stuff. But for now, keep it simple. But it's already complicated, right? I'm sure you guys are confused. Aren't you? How many of you are confused? Raise your hand. Oh my goodness. Maybe you should go back to class 1. Why are you confused? Too much happening. I teach all of this in 1 hour, you know that? In my PhD students in my university. I literally teach epigenetics in 1 hour to people. Okay. We've got 6 days to teach the same thing again and again. All right, at the end of the day, if you guys get something, that's good. That's why it's important for me to ask you questions and you guys have to speak up. What is it that you guys are confused about? Let's ask that before I move forward, because I'll confuse you more. That's what's going to happen. Everybody raise their hand. Every single one of you raise your hand that you're confused. Am I providing you too much information? Huh? I thought I'm repeating the same thing again and again, no? Okay. I'll tell you a story just for fun. So I was there 2 days ago in Haryana. A numerologist came and sat next to me and said, I can tell you everything about you. Just tell me your birth date. I told him my birth date. He says, your brain works faster than everybody else's brain. He asked me the other question before that. He said, what do you do for profession? I said, I'm a scientist. He says, your brain works faster than everybody else, because he knows my birth date. So that's a joke for the fact that I'm thinking that I'm explaining to you everything and you guys are not getting it. And I'm like, okay, stop. Think 5 steps back. My numerologist was helpful to me. Okay. Anyways, what is it that you guys are confused about? What was the most confusing part? The last slide? Let's forget the last slide. What else?
Everything is happening all at once. So like, one part of it is activating it, the other one is repressing it. So all of it is happening at once. So it is difficult to keep track of.
It doesn't happen same place. Right? That's what I'm telling you. It happens on certain genes, because you want certain genes to be expressed, you want other genes to be repressed, you want neurotransmitter to be repressed, LDID to be expressed. But there are like 5,000 such genes as LDID and 5,000 other genes like neurotransmitter. Both need to go through the same thing. Half of them have to be repressed, half of them to be activated. Right? The genome is a beautiful thing, guys. 23,000 genes we got to deal with. Right? That's why you are you. That's why you're not E. coli. Right? I mean, that's true, isn't it? That's why you're not COVID-19 virus. Otherwise, with 30 genes, you would have done so much, right? Anyways, so why am I saying this? Why am I showing you this? Because I want you guys to learn epigenetic patterns in the cell, okay? Epigenetic, I had to show you the complexity so that you can understand, you can appreciate the complexity, get some of it, and get the basic concepts. Major concepts being some things like H3K4 trimethylation is going to be, well, that's what it shows, but I teach you something else up here. It has to be at the TSS. Did I not tell you that? I told you that, right? Transcription activation. Marks genes for transcriptional activation, so it has to be a transcription start site so that it can recruit the Tata binding proteins, remember? Okay, good. It also can be at CPG islands that are unmethylated, okay? And it will just mark that for silence, because if you have methylation, you don't want that gene to be expressed. That's suppressing it, right? So it will only bind to things where it's unmethylated. Easy peasy. Lots of different ways of controlling things, right? That's all. K-acetylation, histone acetylation, on the other hand, you see, follows the exact same pattern. I told about histone code, right? What does acetylation do? Bromodomain proteins, nucleosome remodelers is needed. Both of them are needed, otherwise the transcription won't go on. I said this. Did you guys remember this? Yes. No. Do you guys remember this? Right? Histone code, acetylation, methylation, K4 methylation together. K9 methylation, forget about that. Don't go in there because you'll be more confused. Let's not imagine you didn't see the blue line. Okay? K36 methylation, K79 methylation are on the gene body. They will usually, almost always, are on the gene body. Okay? And they're only on the gene bodies where RNA polymerase has gone through. So they're only going to be on transcribed. So K36 methylation and K79 methylation help RNA polymerase go through. And remember, RNA polymerase, that you've gone through this already, you don't need to transcribe it again. Otherwise, RNA polymerase will not know that, right? It will keep on making. How am I supposed to stop at 1,000 transcripts and not at 10,000? Because I need some memory of transcription, and that memory happens by K79 and K36 methylation. I didn't go into this, but I'm just telling you that now. Okay? Is that cool? That's very cool. The cell is so amazingly brilliant, right? That I need to know. I made only 1,000 copies of this already. I don't need to do anymore. K79 and K36 methylation levels correlate directly with how many transcripts have you made on the gene. If you look at correlation, and you know correlation, have you guys heard of biostatistics? Statistics, R values, R square. So if you do a mapping of RNA transcripts and you do mapping of how many K36 copies are there, and you do a action plot, you're going to get very, very good correlation, 0.6, 0.7, something like that. Okay? Amazing, right? Cell knows how to tell the transcription machinery that I've gone through this. Okay? So that's why they will only be there on the gene body. You see on top gene body? That's where they are. They're not there here. They're not there. They're very specific beauty of it. That's why I do epigenomics. I love it. This is beautiful. How did the cell figure out exactly where to stop, exactly where to begin? On a nucleosome, nucleosome is everywhere, right? Isn't it? And then now look at the red marks, right? K9 trimethylation on the silent genes, gene body, promoter, everywhere. Just covers it up. Says, no way, you're not going to go through. No way. Okay, you see this broad pattern? This is called broad marking. It goes for several hundred kilobases or even megabases sometime. So that's repression for you, gene repression. We talked about gene repression, right? All of it. Is that clear?
Yes, sir.
Okay, so that's permanent silencing. That's genes being silenced by K27 or K9 trimethylation. Whereas transcription start site and the K4 methylation and K-acetylation is very narrow. It's right at that nucleosome where it should be. That's it. It tells me, here is my nucleosome, 200 base pairs. That's where only this thing is going to be, okay? Whereas my marking of repression is like, boom, broad. It can be everywhere, okay? Cool?
Yes, sir.
Got this?
Yes, sir.
So that's why epigenetic patterns are so amazing, guys. I love this. Data. What you see on the screen. This is from my lab, from a melanoma patient's biopsy tumor. Okay, what do you see? Let's see. Let me hear out you guys. What do you see on the screen now? You can say, I see blue. That's fine. You can stand up. Let's interact a little bit for 5 minutes. Yeah.
Various histone modifications on chromosome.
There are various histone modifications listed on the left side. Very nice. They are telling you the track that it belongs to. Remember IGV? Yesterday class I started showing you IGV. This is an IGV picture, guys. The gene is right by the bottom, the one I was showing you. I didn't show you any of this top. I only showed you the gene yesterday. If I opened my IGV and I put this histone modification patterns, this is how it will look. Now you see, what else do you see? Come on, stand up, tell me. Let's interact a little bit. Sing a song together. Great, you said peaks. I love that word, peaks, right? Great, peaks. What does peaks mean in this context?
Higher concentration of marks.
Higher concentration of marks. Good, thank you. So there are peaks. There are peaks like this. Boom, boom, single peaks. Very nice. Narrow, clean peaks. Okay, I told you that. So I told you before this, K4 trimethylation, beautiful, nice peaks, right? K4 monomethylation, beautiful. K27 acetylation, beautiful, nice peaks. When I say K something, it's all usually a histone H3, so I'm not really saying histone H3, okay? So we just ignore the H3 part for a second, but it's all histone H3 for now. Yeah. All right, are we good? Okay, look at K27 trimethyl. It was broad. It wanted to be broad. because it wanted to silence any region around the gene. We got 3 or 4 genes here, 3 genes on the bottom that are actually only some signal in canine trimethylation, but they're mostly silent. This is real data. This is really what is happening in a melanoma patient. Okay? What I showed you before this was just pictures and pictures anybody can draw, right? But I'm showing you real stuff so that you can correlate. That's what happens. Okay? Cool? This is the data generated by one of my postdocs in my lab. We have lots of such data in my lab. We have 4,000 ChIP-seq data sets, and I'm going to teach you ChIP-seq in a second. Okay? But this is ChIP-seq data sets. All right? What does ChIP-seq even mean? We'll come to that. But I wanted you to appreciate in the real world, what I show you is actually happens in the real world. Okay? It's a real thing. What I am amazed with when I see this picture is like, boom, how did it know what to stop right there? Right? How did my K77 trimethyl decide right there I need to stop and allow the rest of it to go through and then stop again? Right? Brilliant. Cells are the most creative part, right? Within ourselves, every single cell, 39 trillion cells, are amazing engineers right inside you, kicking every second for you to comprehend what epigenetics is, right? It's right now. Okay? This is brilliant stuff. I love it. This is where the biology is, guys. Okay? So I showed you something, and I'm showing you a cartoon again, and I'm cartoon again to reinforce the same point that I reinforced earlier. Okay? You shouldn't be confused. There's nothing confusing. K4 monomethylation, I told you marks enhancers. When K4 mono has K27 acetylation on top of it on the same nucleosome, that's an active enhancer. What does that mean? It's gonna fold on to a gene and make that gene active, right? That means transcriptionally active. Okay? So I call this a chromatin state number 1. For lack of better words, I'm not very creative. So I said, okay, chromatin state 1. Okay? Cool? Oh, in the previous one, okay. I also had K79 methylation in the previous one. Did you guys see that? That I didn't talk about. You see K79 methylation? Right on these genes, I told you 3 genes. 1, 2, 3 being transcribed. Beautiful. K79 is right there, but not there anymore because this gene is not transcribed because I'm silent. K79 methylation tells me the same thing as RNA transcription, right? Got it? Okay, good. So K79 methylation is here. K4 trimethylation is here. K4 trimethylation is right where the gene transcription starts. And K79 methylation goes downstream and says RNA polymerase has gone through this thing. K27 K9 trimethyl, I'm going to always call that heterochromatic mark. Heterochromatin is where our repeat elements are, which are silenced by K9 trimethyl. I'm going to call it K27 trimethyl polycomb silencing for a lack of better word, because polycomb silencing happens mostly on genes, but will also happen sometimes on repeat elements. So it also happens on heterochromatin, but a lot of it happens in the genes. Okay? And this is what I ended yesterday's picture with, and I will keep going it because I love this stuff. Okay? Not for anything else. Let's see if it plays. It probably will not play. I hate this. But you guys remember this from yesterday? No. It's the same story, but I've told you, right? You can go through the genome. I can keep looking at it all my life and feel amazed and amazed and amazed every single time because I've got a red bar here. Red is equivalent in this case to K4 trimethylation and K27 acetylation together as a chromatin state 1. Remember chromatin state 1 or 2 or whatever, some chromatin state. Okay, so my red is a chromatin state. Okay, my yellow is another chromatin state. My gray is another chromatin state. My green is another chromatin state. That's my epigenome for you. Okay, my epigenome is marked in different colors, and after using a machine learning tool, so AI again to machine learning and deep learning equals to AI. Okay, all right. So we got this right at this gene. You see where my laser is? See my laser? You see that's an active promoter. It's marking this gene right here and this gene right here. No, not this gene is not going. This is only marking this. This gene is actually silent. You see this between the two? Gray is silent. this gene is actually not firing because there's no green. I know that there's no green, so there is no firing, right? The green is here, so this gene is firing. There is transcription start site here, so this gene is firing. There is one here, so it's firing. Okay, same. So this whole region is firing, not firing, firing, not firing, and there are intermittently there are enhancers. Is this enhancer to this gene? I don't know. Okay, this gene could be to this enhancer too, because it could fold. Remember, we told about folding. For all you know, in the 3D chromatin structure world, all these reds are actually at one place in the 3D chromatin. If I am this, maybe there are